Okay, so I'll demonstrate to you how to perform microculture. So what are the materials needed for microculture? So you have your inoculating needle, then your inoculating loop. Okay, so you have your inoculating needle, your inoculating loop. Ginaben ako siya nga inoculating loop. This one will be used in transferring the small portion of your agar to your microculture setup. Okay. Then next you have your forceps, then coupling jar with alcohol, distilled water, sterile distilled water, lactophenol cotton blue, slides, cover slips, and your SDA or subarodextrose agar. This one. Okay. So first, another is you need a sterile petri dish, clean sterile petri dish. Okay. So let's start with the microculture setup. So first is you need to open the plate and place two applicator stick or toothpick. After that, get a glass slide and sterilize this one using flame. Okay. Then put it in your microculture setup. So there are two layers now in your microculture setup. First, you have the toothpick, and next level, you have the glass slide. Next is you need to transfer a small portion of agar to your uh, microculture setup. So you have your SDA. Next is to cut a small portion, okay, a square, ng agar, okay, of the size of that agar will be 1/2 by 1/2 cm. So we'll use a cover slip to cut the agar. Okay, so first is we need to sterilize it and cool. And cut approximately 1/2 by 1/2. Okay. So next is using a bent inoculating loop. We'll transfer the agar from this original plate to your microculture setup. Cool the inoculating loop, bent inoculating loop. Next is transfer the square na SDA from the original plate to your microculture setup. Okay. Next is we need to inoculate our sample. So using the needle, okay, sterilize the needle using a sterile needle or sterile loop. Okay, inoculate your specimen. This is our specimen. So we need to pass this. Then touch the colony, scrape. Okay, then after, inoculate this one to the four sides of your agar. 1, 2, the second side, third side, then fourth side. Okay. Disinfect or sterilize. We use inoculating needle. The next is put a cover slip. Okay, we'll use this cover slip. So sterilize. And next is to place this one over the agar. Okay. So for your setup, you have your applicator stick, cover slip, your agar inoculated na, then cover slip. Okay. Then after that, get a sterile cotton ball or filter paper, and wet it with water, approximately 10 drops of water per cotton ball. Okay, so place the first cotton ball here, the wet cotton ball. Okay, and the second wet cotton ball here. Okay, so the purpose of that wet cotton ball is to give moisture to your setup, to your microculture setup. Then cover it and incubate this one for approximately 7 days. Okay, or until nanay spore formation, or if the colony is matured. Okay, so after maturation or after the incubation, next is harvesting phase. So how to harvest the microculture setup? So open this one. Then first is get a clean glass slide. Sterilize a clean glass slide. The next is place a drop of LPCB. Okay. The next is get the cover slip from your microculture setup. Okay, and use this one as source of specimen, or this cover slip contains the fungal element. Okay, so drop. Okay, and you have your first preparation. So wipe the excess stain, and next is to seal this one using nail polish. Okay, so this one is the first uh, product of your microculture. The next is to remove the uh, remove the uh, agar using bent inoculating loop and your forceps. Remove this one from the setup. Okay, so and discard this. Okay, so ano lang nato ibutang. Discard. The next is to get the glass slide. Okay. And next is to add a drop of LPCB to the area with fungal element. Okay, one drop lang. Then next is get a clean cover slip. Sterilize it. Okay. And next is put the cover slip. Okay. And you have your second preparation, slide prep from your microculture. So you have two slide preparation from your microculture setup. Okay. So wipe the excess stain. Okay. That's why you need to use only one drop of LPCB to avoid excess amount of stain. So after wiping it, next is to seal it using nail polish. Okay, so that's for your microculture. Okay, so those are the important steps for the whole procedure for your microculture.